Today I'll be talking about developing an improved understanding of the structural function of the wheat root microbiome. So I'm Tim, and I'm at Rothamsted Research. So by way of an outline, I'd just like to go through these points here of actually what the talk will be about today. So firstly, I'll just give an overview of what is the plant microbiome, how we study it, why we study it, and then what I'll do is move on to a few case studies about the study of the microbiome, the root microbiome in particular, and this is to do with the effect of nitrogen fertilisation on the bacterial wheat root microbiome and the effect of land management on the bacterial wheat root microbiome. And then finally, there'll be another case study to do with the effect of dwarfing on the root microbiome. So this is in relation to plant breeding. And then those three case studies are pretty much culture independent methods that they're based on. So the fourth, another part then is to actually talk a little bit about culture dependent methodologies, and that's using Pseudomonas as plant growth promoting rhizobacteria. So what is the plant microbiome? So the way I like to describe it is it's the complete microbiome, the complete microbial community that's associated with the plant. And then we can divide this into above ground and below ground components. In terms of the above ground components, we can think about the phylosphere mainly, which is the microbes that are associated with the actual tissues that are above ground. And then below ground, we can think about the rhizosphere, which is the soil which is closely associated with the root system. And then closer to the root system again, we can think about the rhizoplane. And the actual root microbiome, which is mainly composed of those two elements, is generally selected from the bulk soil. And then in addition to the rhizosphere, the rhizoplane, and the phylloplane, we can also consider the endosphere. So this is the microbial components that exist within the actual plant itself. So, but generally they exist between the plant cells. So they're not so much intracellular, they're more intercellular, with the exception of some symbionts, mutualistic symbionts such as rhizobia. And the sort of organisms that make up the microbiome are bacteria. That's a large proportion of the microbiome. There's also fungi and archaea, but there are other members of the community as well, such as oomycetes and nematodes. So how do we go about studying it? Well, this slide here is just to describe the sort of culture independent methodology, and this can be done for two main ways. So we can do this using this 16S based approach. This is for what we call amplicon sequencing. And so this is where we would take the community sample, we actually extract the DNA from it, amplify this DNA, and then perform analyses to actually get an understanding of the structure of the microbial community. And of course, this doesn't have to just be based on 16S, it's whatever gene that is amplified. And then in addition to that, there's also another approach which still involves using the microbial community sample, extracting the DNA from that, and then performing shotgun metagenomics. And the key difference between the shotgun metagenomics and the amplicon approach is that the amplicon just considers a small compartment, if you like, or a small gene sequence in the actual microbiome, whereas the shotgun metagenomics is able to look at the whole gamut, if you like, of the genome of the microbiome. And so essentially, we're able then to do some analyses on this data sets that come out of this, and we can look at the relative abundances of the microbial communities. And we can also look at the relative abundance of their particular genes which are involved in different functions. So there are two main approaches there for studying microbiomes for using a culture independent methodology. And then moving on to another type of approach is what we call culturomics. And this is essentially where we actually would look at the cultural part of the microbiome. So this has been done sort of quite in depth for the gut microbiome. And we can see here in this figure here that the actual overlap between the culture-based retrievable microbiome and that of the culture-independent methodology occurs. But there are also elements that are in the culture-independent approaches which are not picked up in the culture-dependent approach, and vice versa. So this just goes to show that both methods are important. And one of the reasons we don't always capture the full microbiome component, if you like, in the culture-based method is that it's actually quite difficult to culture all members of the microbial community, because they all have, or some of them have, quite specific requirements for growth. But what we should say here is also there's been some studies done in Arabidopsis here where we can actually see there are actually quite clear overlaps in the ability to culture microbes along with the total community. And this is one approach that we're going to be talking about today, which I will touch on later. So why do we actually study the microbiome? Well, we believe that the soil microbiome has potential for contributing to more environmentally benign agriculture. And it's well known that naturally occurring soil-dwelling microbes can influence the plant health, growth, and resource use efficiency. And there are well-known examples. I touched on rhizobia just a moment ago, but in addition to rhizobia, there are other microbes which can be beneficial to plants, such as arbuscular mycorrhizal fungi. But we think there's a lot more to the microbiome than just those two examples, in that they can actually, there's a broader component of the microbiome which we think can be beneficial for plant health. And we feel that using recent advances in nucleic acid sequencer technologies, we can actually start unearthing these microbes and actually starting to actually elucidate their functions, and together that would then give an opportunity to better understand and exploit this resource. So then, to move on to the first case study, this is to do with nitrogen fertilisation and the bacterial root microbiome. So why do we actually want to optimise nitrogen use in agriculture? Well, to cut a long story short, really, we want to just optimize the yields for sustainable production. We want to actually make agriculture more benign. We want to reduce losses from denitrification and leaching. And when we consider that 75% of nitrous oxide emissions come from agricultural soils, there's some work to be done there to reduce the impact of nitrogen on climate change. So what we've done in this study is we've looked at one of the experiments at Rothamsted called Broadbrook. It's probably Rothamsted's most famous experiment. It was established in 1843. And there are lots of different parts to this experiment, but today I'd like to focus on the continuous wheat. So that is that's been continuously cropped with wheat since 1843. And this figure here, you can see the different treatments that we're going to talk about today. And this is FYM, is farmyard manure. This is no inputs at all. Then we have medium and high inorganic nitrogen inputs. And so all we simply did in this experiment was to actually look at the bulk soil and the rhizosphere of these of these plots where these nitrogen treatments have been added. And we did sampling at both a 4-week time point as well as a 10-week time point. And the 10-week time point was at plant flowering time. And what we found was quite clearly, looking at the bulk soil plot here, which there was very clear differences in the communities according to the fertilisation regime. And there was no difference, though, between the actual two sampling time points. And then when we moved on to the rhizosphere, what we could see again, that was all plots again separated by treatment. But furthermore, we were able to see that 3 of the 4 plots also separated according to plant growth stage. So this was at the 4-week stage and the flowering stage, and we could see a clear temporal separation. But this wasn't the case for the farmyard manure. So we can see for some reason this is a more stable system in terms of the community dynamics. And this could just be, there are various reasons this could be, but one reason could be that the actual addition of the organic matter into the farmyard manure could actually contain components of which are members of the microbiome. So in terms of what bacteria are responsible for the community shifts, the sort of things you can see according to this plot is that we found that the actinobacteria and the proteobacteria were always abundant regardless of N treatment. We found that the Bacteroidetes responded to high levels of inorganic N, whereas we found that the acidobacteria, the testibacteria, and the planktomycetes were less competitive under inorganic N treatments. So just to summarize that small part about the nitrogen work, you can see that the bulk soil bacterial populations are distinguishable by N treatment, and the rhizosphere bacterial populations are also distinguishable by N treatment, but there's also an added temporal stage here to do with plant growth. So the next part I'd like to talk about is bacterial diversity in the Highfield wheat plots. So Highfield is another one of Rothamsted's long-term experiments, and I was going to spend a few moments to describe this experiment. So essentially, the Highfield site prior to 1949 was completely grassland. And then in 1949, it was divided into different land uses. So firstly, some of it was converted to arable, some was maintained as grassland, and some was converted to bare fallow. But as you can see, that bare fallow conversion took was a bit later than 1949. And then from 1949 or 1959 all the way to 2008, these different treatments were maintained. And then in 2008, these treatments were then further subdivided. So one example here is that the arable was then subdivided into bare fallow, grassland, and some of it was maintained as arable. And this combination, if you like, happened for all of the 3 treatments. So in addition to the grassland, plots were then converted to arable, bare fallow, and maintained as grassland. And really, so what we wanted to do in this experiment was to look at the effect of land use, and does it alter the bacterial diversity in the bulk soil and the rhizosphere. And we focused our efforts here on the wheat plots. So first of all, we just wanted to look using an amplicon sequencing methodology. What is the phylum level discrimination between the bulk soil and the rhizosphere? And we could see quite clearly, according to this here, that there were differences in the abundance of the Bacteroidetes as well as the proteobacteria and the actinobacteria, with all of those being more abundant in the rhizosphere. But the same as the experiment at Broadbork as well, we found that there were some phyla which were more abundant in the bulk soil. And you can see here this examples such as the acidobacteria and the verruco microbia. So then when we actually looked at the ordination of these 16S data, we could see that there was a clear separation of the actual the actual microbiomes according to niche. And this was so clearly in this situation here, we see the rhizosphere as opposed to the bulk soil. But there was also a sub-niche level discrimination, which was based on land use. So this was, as you can see, according to here, if we look at the rhizosphere, that there's the main separation between the rhizosphere and the bulk soil. But then within the rhizosphere plots themselves, we can see separations according to the land use treatment. So to summarize that part of the talk, we can see that bulk and rhizosphere communities are distinguishable. The proteobacteria, Bacteroidetes, and the actinobacteria are more abundant in the rhizosphere. The acidobacteria, verrucomicrobia, and the gemmatomites and the planktomycetes were all more abundant in the bulk soil. And so what we're able to see here is that the rhizosphere and bulk soil communities are distinguishable based on land uses, but there are differences, and these are more subtle, and there are shifts at the phylum and class level between both bulk soil and rhizosphere samples. Okay, so I wanted to move on to the third case study in terms of the culture-independent methodology. And this is the effect of dwarfing on the bacterial wheat root microbiome. So by way of an introduction to this part, before the Green Revolution, wheat plants were very tall. And because of this, the stems were often not strong enough to support the heavy grains. And so in a process called lodging, the plants would fall over. And then because of that, there would actually be an increased risk of fungal infections and increased problems with harvesting. And essentially, there was a decreased production and associated economic losses. And so as part of the Green Revolution, there was efforts to breed wheat plants that were high yielding and were shorter, and they also responded well to inorganic fertilizer application. And the reason that this was achieved was through the introduction of the RHT genes, or mutant alleles of the RHT genes, into wheat. And what this actually results in is a reduced stem internode length and generally a smaller, shorter plant with reduced sensitivity to gibberellin. So essentially, the RHT genes that are in the dwarf are resistant to gibberellin, so that actually then suppresses stem elongation. And then as a consequence of this, a higher proportion of the photosynthate is partitioned to the grain and increases the number of grains with the spikelets of each spike. And ultimately, putting all this together, so when you add lots of fertilizer with these different dwarf genotypes, you end up with increased yields. But at the time, there wasn't really any consideration about how the breeding process is affecting the microbiome and the bacterial diversity in the rhizosphere. So what we did in this experiment is we looked at some tall cultivars and some short cultivars. And we did some amplicon analysis based on the 16S. And what we found was that the structure of the bacterial communities from the tall cultivars was statistically different from the semi-dwarf cultivars. In addition, the tall cultivars tend to have a lower diversity than the semi-dwarf cultivars, which was a little bit surprising. We were expecting the tall cultivars to have the higher diversity. So you can see here, according to this ordination, there's very clear separation according to plant height, with the semi-dwarf here and the tall here. And in addition, as I mentioned there, looking at the observed OTUs, as well as the Shannon diversity indices, there's actually a higher observed OTUs in the semi-dwarfs compared to the dwarf and the tall, sorry. And there's also a greater diversity in the semi-dwarfs compared to the talls. And so what we wanted to look at next was, what's actually causing this change in structure. What is the difference between the microbiomes of the tall and the semi-dwarf cultivars? And what we found when we looked at the differential abundance of across the two different types of wheats here is that the proteobacteria, the Bacteroidetes, and the actinobacteria are differentially more abundant in tall cultivars. And we also found that the verrucomicrobia the acidobacteria and the planktomycetes are differentially more abundant in semi-dwarf cultivars. You may remember from some of the previous slides that what we found is that in the rhizosphere, in the previous experiment, we mentioned that the actual Verruca microbia, acidobacteria, and the planktomycetes are generally more abundant in bulk soil, whereas the proteobacteria, Bacteroidetes, and actinobacteria are actually more abundant in the rhizosphere. So it seems like there's that the semi-dwarf microbiomes are more akin to the bulk soil, whereas the tall cultivars are more akin to rhizosphere soil. So this, if we also think about the potential impact of gibberellin insensitivity and the fact that gibberellins were first discovered in microbes, that the insensitivity of plants, the semi-dwarf plants, to gibberellins could actually result in a reduced amount of crosstalk between the microbiome and plants. So what we did as well was looked at the networks of did some network analysis looking at the talls as well as the semi-dwarfs. And what we found was that the tall cultivars were much better connected. They actually had a much greater amount of module hubs here, as well as connectors. Whereas if we look at the semi-dwarf, they have a small amount of module hubs here and connectors. So even though the tall cultivars seem to have a lower species richness and a lower diversity than the semi-dwarf cultivars, they actually seem to be a better connected microbiome. So the next thing I wanted to do was tell you a little bit about some of the shotgun metagenomics that we've been doing. So up until now, the work with the microbiome and the wheat cultivars has involved amplicon sequencing. And I also mentioned the hypothesis that the bulk soil, or rather I should say, is that the dwarf cultivars seem to have a microbiome which is more akin to the bulk soil than the tall cultivars. So what we actually did in this experiment was include some bulk soil controls. So looking at these two ordinations here, this is the amplicon here, and this is the shotgun metagenomics. And we're broadly seeing the same trends in that we're able to see clear separation of the microbiome according to plant height. So on this left-hand figure here, you can see that the reddish dots are all the tall cultivars, and the bluey greens are the short cultivars. And what we're able to see here is that there was actually a similar separation according. This is, again, just to reiterate, this is the amplicon here, and on the right-hand side is the shotgun metagenomics. And we see here that the same sort of pattern, but crucially, this is the bulk soil here, these grey dots here, and they are more similar to the semi-dwarf. So this hypothesis that the microbiome of the semi-dwarf cultivars is more similar to the bulk soil than the tall cultivar seems to be correct. And then the other thing I wanted to draw your attention to was this figure here. And so the shotgun metagenomics is, as I mentioned earlier, is able to take into account more than just amplification of a specific gene sequence. We're able to look at the different functions or functional genes in these systems. And the genes here, which we're seeing, are either differentially more abundant in the semi-dwarf or the tall cultivars. And essentially what we're seeing in the tall cultivars, I'm sorry if you can't read a lot of this because it's quite small, but what we're seeing is that there are a lot more transporters, especially ABC transporters, so genes for transporting of nutrients, for example, secretion systems, and transcription factors, as well as genes involved with quorum sensing, are more abundant in the tall cultivars as opposed to the semi-dwarf cultivars. So this again is supporting this notion that the crosstalk, if you like, not just between microbiome and the plants, but also between the microbiome with other members of the microbiome, is more prominent in the tall cultivars opposed to the semi-dwarf cultivars. So I'm just going to bring to an end this part of the work, really. And so this was just to say that the tall and short wheat microbiomes can be discriminated. The short cultivars have a higher diversity but less complex community networks. The short microbiomes are more similar to the bulk soil. Tall microbiomes have higher proportions of PGPR-like taxa and a higher abundance of transport genes and communication genes. And then what we're doing going forward now is to examine ancestral wheats with as well as RHT mutants with altered sensitivity to gibberellins. So these are RHT mutants in an isogenic background, so we can really zoom in and see if this gene is highly important. But including the ancestral wheats, we're able to go back in time, if you like, to look at the very primitive grasses which are related to wheats and all the way through to our modern elite cultivars. And then another key element we're developing at the moment is to examine the interaction of plant genotype and fertilisation regime. So we've been talking quite a lot about nitrogen and genotypes. So it really is to bring those two elements together. So there's a lot of work out there looking at amplicon microbiome analysis, but what does this really mean? So I think the important thing is to firstly develop an appreciation of the players in these systems. But really, the amplicon work doesn't really give much of a lead on the function of microbes. And as you saw just on one of the previous slides, there was some metagenomics work there as well. So we need to do more of that sort of approach, but also we need to layer that with other approaches such as culture-based transcriptomics, metabolomics, as well as proteomics. And then once we've actually brought all of that together, we'll get a better understanding of the rules of engagement between hosts and the microbiome. And hopefully we'll be able to manipulate the systems to optimize microbiome function. And then once we understand all of that much better, we'll be able to develop inoculation strategies, potentially improved soil amendments, as well as the amendments of plants for recruitment of microbes through root exudates, for example. So, as I mentioned, then we need to actually develop our understanding of the function of microbes. And one way I wanted to explore that is to use a cultural-based approach. And so what we've been doing at Rothamsted is to use Pseudomonas fluorescens as a model organism. And we think this is a good system. It's amenable to culture, and therefore we can do phenotyping of it, mutagenesis, and genome sequencing. And the fact that it's also metabolically diverse means that we can actually look at how tie together the biology and the chemistry. And we also feel that it's a good organism to work with because it's antagonistic to certain pathogens such as take-all there, but there's also Septoria fungus as well. So there are three elements I wanted to touch on in terms of Pseudomonas work, and this is the different abilities of bacteria to actually perform functions which are beneficial to plants. So firstly, I'll talk a little bit about abiotic stress relief with the example of ACC deaminase. I'll move on to nutrient acquisition, both macro and micronutrients. And then finally, a small part on disease suppression of Septoria and a pipeline that we're developing for that. So yeah, firstly, to move on to the ACC deaminase study. And this was done at the Highfield Experiment at Rothamsted. So just as a reminder, the Highfield experiments, we have these different land uses. So what we did for this particular study is we collected soil from the continuous arable, continuous grassland, as well as the continuous bare fallow, brought that back to the lab, grew wheat plants in that, and then isolated pseudomonids from the rhizosphere, the bulk soil, and the roots of these plants. And then what we did as well is looked at the ACDS abundance and also looked at their phylogeny through the GIA-B gene. So what is ACC? Well, it's aminocyclopropane carboxylate, and that's the deaminase part is actually the ACDS gene is ACC, encodes ACC deaminase, and that converts ACC, it converts ACC to alpha ketobutyrate and ammonia. And the potential benefit of this to the plant is that it actually stops the buildup of stress ethylene. And even though ethylene is important for plant development, too much of it can be a bad thing. So the control of ethylene release is an important way that microbes can actually benefit plant health. And so what we did is we looked at the different the ACDS phylogeny and abundance across the different land management types, and we could see that there was a difference in the amount of isolates, and they were more abundant in isolates from the bare fallow as opposed to the arable and the grassland. And we could also see that there were some phylogenetic differences according to the isolates and where they actually originated from. So just to reiterate, we found that there were less in the bulk soil, and as you get closer to the plant itself, the abundance increased in isolates which actually housed the ACDS gene. So we also wanted to look into to check the function of these bacteria, as opposed to just assuming that having the ACDS gene actually proves function. We actually did an assay to check this was the case. So this was using this standard curve here. We're using ninhydrin. And what we're able to see is that using, if we can look at this graph here, this is showing the utilization of ACC as a sole nitrogen source. And what we could see is that compared to the negative control, which is the red line here, which wasn't able to grow on it at all, isolates which were derived from the arable system, arable soil and the bare fallow soil and the grassland soil, which had the ACDS gene, were able to grow on this as a nitrogen source. But crucially, what we noticed as well, that there was a differential ability of isolates to do this depending on where they actually originated from. So we can see here that the isolates from the grassland soil which had it were able to do it, but they were less capable compared to those from the arable and the bare fallow. And this graph here just shows the ACC consumption. So again, we can see here that the negative control is unable to consume ACC, whereas the other isolates which have the ACDS gene are able to do this. But yet again, the grassland isolates are less efficient at doing this compared to the arable and the bare fallow isolates. So to summarize that part of the work, what we're able to see is that land management as well as niche influence the ACDS gene abundance in Pseudomonas. And the ACDS function is less efficient in the grassland soil. And is that so I say healthy ? Soil. So it's a potentially a health indicator gene that we could use and develop to show that actually in healthy soils there is less ACDS gene abundance. So I'd now like to move on to another part, which is to do with plant growth promoting bacteria and nutrient acquisition. So this is based on a very simple experiment where we essentially grew wheat with and without fertilizer. And so we were able to look at two niches, and we looked at the rhizosphere and the rhizoplane. And see here we started with about 1,500 different isolates in the experiment. And what we were keen to do was look at the impact of chemical fertilizer on plant growth promoting rhizobacteria in wheat. And so these nice assays here just show the ability of these microbes to actually perform these functions. So this particular slide here, we've got iron, so siderophore production, potassium, and phosphorus. And if you see here, these are non-fertilized wheat, bacteria from non-fertilized wheat, and this is from fertilized wheat. And there was a clear difference in the ability of the microbes to actually perform these functions depending on where they came from. So essentially, isolates that came from the fertilized wheat were far less able to perform these plant growth promoting functions than those that came from the non-fertilized wheat. And we found that this was the case in that, but soon again, the same as with the ACDS system, that's actually when you get closer to the plant root system, this effect became more pronounced, to the extent that 91% of the bacteria which were isolated from the non-fertilized samples displayed at least one PGPR function, and then in the fertilized samples this number was actually 19%. So there's a huge difference in the ability of these microbes to perform functions depending on whether the plants have been fertilized. Okay, and then the final case study here is to do with Pseudomonas bacteria, again, for the control of septoria. And so just as an example, a way into this, septoria is an important wheat disease, and it's a big problem for UK as well as the rest of European wheat farmers, and it's caused by Zymus septoria tritzi. And fungicides traditionally used to control this disease, but however, resistance is being evolved, and we're thinking about developing new alternative strategies to actually control this fungus, and we think that the plant microbiome could be a good source to do that, either as biological control agents or as a source of natural products. So what we've been developing at Rothamsted is a natural product and biocontrol agent discovery platform for the suppression of this pathogen. And the way we've gone about doing this is we've developed a suppression screen here, where we actually are able to culture the fungus in its yeast-like growth form, and then we will then challenge the fungus with bacterial cultures. And you can see here that there's a differential ability of these microbes to suppress the growth of the fungus. So these areas of clearing here, these three isolates, for example, as well as these two here, are able to suppress the fungal growth, whereas these ones don't seem to be able to have that function. But the key thing here is we're able to then link together the phenotype platform with a bioinformatics guided approach. And so we can then sequence the genomes of these bacteria. And then with this approach, we can then come up with candidate natural product genes, and then we can then try and prove the function of these. So this is an example we have here is the mutagenesis of the PHLD gene, which is is partly responsible for the production of DAPG, which is a well-known antifungal metabolite. And so what we can see here is that using this example, that we have the wild type, two examples of wild type cells, which are cultures that can actually suppress the fungus, but as soon as we've knocked out the PHLD gene, the ability to do that function is abolished. So we can see that this is a nice platform to actually develop the new candidates for biocontrol or natural product discovery. So just to finish off, to start, when Ajay got in touch with me about this talk, there was some notion that we have to put some learning outcomes. And so these are the three that I'd suggested before I gave the talk. And so the first one is to learn about the factors that affect the root microbiome. So today we've been speaking about the fertilisation, as well as land management, as well as plant genotypes. So there are three factors that are important, but there are many other factors, and I'm sure that future research will unearth those. Then also to learn about the functional potential of the microbiome. So we've been focusing on looking at Pseudomonas bacteria, and we've used it. This has been using a culture-based approach, but as I mentioned earlier as well, we can use other approaches to look at the function of the microbiome. But then in addition to that, we can also see that there are the different types of activities that microbes have, which can be of benefit to the plant. So this is in terms of nutrient acquisition, so the solubilization of nutrients, which ultimately can be given to plants through abiotic stress relief. There, we've touched on ACC deaminase. And then in addition to those two, there's also the ability of microbes to suppress plant pathogen growth. And then the final element here is just that the different methods available, as I've been mentioning, that we can look at culture dependent as well as culture independent methods. Okay, and I'd just finally like to acknowledge the team at Rothamsted. So they're all there. So thank you very much, and also from the sponsors. So thank you very much. Any questions?
Thank you. Thank you, Tim, for today's webinar. And thank you, everyone, for attending today and asking questions. So we already have a few questions. The first question actually came before you presented your slide about shotgun metagenomics. So I think you have answered part of it, but I'll read the question. While 16S is cheap, it is just one gene region, and you can't look into the fungal diversity with this single gene region measure. Also, there are many problems with 16S quantification, as many bacteria have divergent 16S copies or different 16S copy number. Don't you think that whole genome shotgun data might help to gain a more accurate estimation of microbial diversity, especially given falling prices, or at least nanopore whole genome 16S sequencing?
Yes, I agree. Yes, absolutely. I mean, yeah, some of this work was done before the prices have been lowering, but it's still obviously a lot more expensive than doing amplicon. But I think that one of the strengths of amplicon sequencing is that it can give you a way in and sort of inform you of where you should be then doing the shotgun sequencing.
Great, thank you. Next question. Which tool did you use to profile your shotgun metagenomic data? Is a beta diversity presented in the principal component analysis based on only bacteria or other detected kingdoms? Finally, what was the percentage of your reads that you could map to a used reference database? Soil microorganisms are generally not good represented in these databases.
Yeah. Okay. I'm not sure the answer to the second part of that question. How many were matched to but we can find that and get back in touch about that. In terms of what that data is that shows that was bacterial, we've deliberately made the we've got that those data sets, and we were looking at essentially they're very large data sets, and we've decided to focus on the bacteria, and those plots were based on all functional genes. They weren't just taxonomy plots.
Okay, so how many soil bacteria do you estimate are actually culturable? Also, is Pseudomonas species dominant in terms of biomass in this community? Thanks for a nice talk.
Oh, thank you. I think that one paper that I touched on at the beginning, which was an Arabidopsis study, claims that I think it was 70% of the actual root microbiome is culturable, and that study used a whole bunch of different culture methods, and also that was done, I think, primarily on the rhizosphere as opposed to the bulk soil. So I think that we'll probably get closer to culturing the majority of things in the rhizosphere compared to the bulk soil, because of the bulk soil organisms, I think, can be quite picky in the way that they grow. But we do think that if we actually use methods such as dilution to extinction, where we dilute the sample to the point where we can have a single cell in an individual microtiter well, for example, there's a chance of actually culturing. I don't want to sort of commit to say a particular number, because but I think there's a chance that we could actually culture a good proportion of both the bulk soil and the rhizosphere microbiome using the dilution to extinction approach, where the competition is reduced. So essentially, if we're actually growing on a rich medium, I think it's very difficult for some fastidious microbes to grow, because they just completely outcompeted by the more the faster growing heterotrophs. So I think that there are ways and means to try and improve things, I think.
Okay. Do you think that read length and depth of the NGS stack minion PacBio is good enough to have confidence in metagenome assembled genome generated from metagenome shotgun sequencing?
I don't know for sure, but I believe that that it's improving. I think the error rates, from my colleagues tell me, the error rates are decreasing all the time. So potentially, but I don't know for sure, sorry.
Do you plan to study other widely known pathogens and effects of microbes to suppress them, developing multi-resistant plants and strains?
Yeah, I mean, that's a good question, thanks. So what we're doing is we're using the Zymoceptoria assay as a way in, if you like. The reason we like using it is because of this. I mentioned this sort of yeast-like growth form of it. So it's actually quite easy. When you almost grow it like almost like a bacterial lawn, and so it makes it very high throughput. And so what we'd like to do is then test those microbes against other pathogens as well. So at Rothamsted, we're focusing on Fusarium, as well as Tacol, with Zymoceptoria as well. So we're looking at sort of three major wheat pathogens there, and we're interested to see how this high throughput strategy works. Can we actually see if the microbes that are able to suppress Septoria are also useful at suppressing other fungal pathogens? And we think that's likely to be the case. But then another thing that we can also do is use this approach to also look at potentially medically important pathogens as well.
Do you perform some tests in bacteria in vitro or in silico to evaluate biosecurity in growth promoting bacteria?
Yeah, okay. What I'd say is that we do a lot of in vitro tests, and then we actually, our way into all of this is to actually test microbes and their ability to perform a function. And then when we actually have a positive bacterial isolate that can perform a function, we like to then try and examine the genome to actually get candidate genes which are responsible for that function. So, but it's also important to have microbes that can't perform functions as well to be able to do that. And then in addition to that, it's important to have a good depth of microbes that are phylogenetically quite well related. So if you wanted to make comparisons between, so for example, Pseudomonids, it's quite a good strategy, we think, to have microbes that can and can't do a given function, so we can actually then make that comparison. But I'm not sure what was meant by the biosecurity part of the question.
Okay, so next question is, thanks for your presentation. I'm focusing on the soil microalgae. Is it possible to use the microbiome to identify both cyanobacteria and eukaryotic microalgae, and which database, green genes or silver?
I'm not sure about that one, if I'm honest. We're generally focusing on prokaryotes for our amplicon work, but we do some work using ITS for fungi, and but I'm not sure about. I mean, the cyanobacteria, I would imagine they would be okay and actually detectable in using the amplicon approach, actually. So, and I think that just using green genes or silver should be good, I would have thought.
Do you have any results on microbial profile discrimination beyond the phylum level you presented?
We do, yes. I mean, I think that we do present the data in at the phylum level when we can actually see differences at the phylum level. I think it's nice to actually show that, but then sometimes if we're looking at particular samples and then we can't see the differences at the phylum level, then we will pull out ASVs that are differentially abundant between samples. So that is definitely part of the approach as well.
Many thanks for your presentation. I study rice rhizosphere. I would focus on fungi. In some papers, they use for their identification 18S, and in some others, ITS. What is the best approach in your opinion?
We're doing more and more ITS as opposed to 18S. That's what we've done both, but we seem to get better results from the ITS.
What do you think about prediction of microbiome functions through platforms like PigRust?
Yeah, well, actually, we've done that on occasions for some of our amplicon data, and we can definitely see trends which seem to be sensible. And but we need to. I would like to do some more work to compare our shotgun to amplicon work to sort of to confirm that. But I would always say that the shotgun would be better if you're trying to look for function than looking at predictive software. But sometimes budget constraints won't allow that, I guess. Yeah.
Is someone doing intercropping wheat with maize? Is there any outcome of the improved outcome of microbial diversity?
We actually have some unpublished data on intercropping of wheat with legumes, and we can definitely see that there are differences in the community profiles according to the intercropping strategy. But this was a descriptive study, so we don't really know what the potential benefits are of that. We can tell you they're different, but we don't really know, if it's a beneficial outcome or not at the moment.